descrivo un po' il lavoro fatto da Andrea durante la sua tesi di diploma di primo livello. Andrea si è occupato del design, dello sviluppo e della validazione di un metodo di intelligenza artificiale basato su modelli, diciamo, piuttosto recenti, quali i Vit Transformer, accoppiati a reti convolutive un po' più tradizionali, al fine di poter classificare delle TAC polmonari con l'obiettivo di trovare una correlazione tra tumore ai polmoni e espressione genica di questi polmoni. Il problema è abbastanza importante, soprattutto nell'ambito radiologico, perché la presenza, diciamo, delle espressioni di un gene specifico può abilitare diciamo, l'uso di specifiche terapie. Io passerei la parola, diciamo, ad Andrea, perché così vi descrive un po' tutte le fasi del progetto, dal design allo sviluppo fino alla validazione.
Grazie al professore Spampinato. Allora, la parola al candidato Andrea Virgillito, che ha 20 minuti per la sua presentazione. Mi avvicino a lui per...
Okay, thank you. Okay, for those of you who don't know me, I'm Andrea Virgillito, and today I'll be presenting my thesis work, which is a process of next generation sequencing through a Vit-based model and attention rollout, but I'll describe all this work shortly. First of all, my work follows a trend inspired by some recent research in radiogenomics, who basically spotted a high correlation between quantitative analysis of visual data, which is basically whatever can be exploited through computer vision techniques, and such as morphological and texture feature, to genomics information about cancers. First of all, it's absolutely worth mentioning a couple of papers related. So the first one is by Yi Yang, who implemented a 3D segmentation model trained to estimate the volume of lung cancer lesions to quantify immunotherapy effectiveness. And the other one is by Cha Kenny, who implemented this 5-layer convolutional neural network used to distinguish between the bladder lesions in different stages. So in the last decade, so very recently, researchers focused their attention on, in particular, on the influence of some types of genes like the EGFR, the epidermal growth factor receptor, to the effectiveness of some therapies to attack cancers. Next generation sequencing can be defined as more generally as a massively parallel sequencing technology that offers ultra high throughput, scalability, and speed. But the point is that nowadays DNA sequencing technologies struggle to catch up with current genomics demand. So next generation sequencing has revolutionized the world of biological sciences. And today it allows labs to perform tasks at a level never possible before. So biological system can be studied at a deeper level. Those are a series of tasks that can be performed through NGS, such as sequencing whole genomes or scoring novel RNA variants and splice sites, as well as study the human microbiome or identify novel pathogens. This table summarizes a series of both advantages and disadvantages. So first of all, we have the that such tools are cheap, so low price. We have speed and also lack of invasiveness. Indeed, NGS are presented as a valid alternative to the very invasive technique of biopsy. They are used both in clinic environments, but also in research, so they are freely available by the average researcher. And also, they show a high availability of commercial platform and specialized kits. But we have also a bunch of disadvantages, like for instance, the need for specialized software and analysis, Also, there's no standardized material for clinical application. So there's no fixed ready-made path to be followed for each case, but each scenario should be treated on its own. And third, this technique might still be too expensive for some developing countries, so the poorest one. As you may know, lung cancer is still one of the deadliest and most commonly diagnosed type of cancers. And the high death rates are mainly because of two reasons. The first one is that lung cancer is typically shows low response rates to therapies, and the second one is that lung cancer typically is spotted when it's too late, and that's the reason why previous and ongoing research studies are focusing on discovering useful and reliable biomarkers for the diagnosis of lung cancer and also of its prognosis. Indeed, scientific literature sustained the thesis that says that there's a high correlation between lung cancer tumor and specific mutations in the DNA. And also, it's worth mentioning that though the traditional therapy to attack lung cancer is chemotherapy, patient can benefit from the design of targeted therapies that can be designed using exploiting information from visual data and genomics. So computer vision really helps in doing this. Okay, here comes the artificial intelligence part. So I will be presenting a vision transformer based model. So I would like to spend some words about what a transformer is. First of all, the transformer architecture was firstly introduced in 2017, and it was presented as the great successor of all those models based on long short-term memory blocks and gated recurrent units. The idea behind such models were to, for instance, analyze a sequence of inputs, like, for instance, it might be a series of words in Italian forming a phrase, in order to perform tasks like translate to this phrase into an English one. The problem was that they were not so good in achieving this. The idea was keep collecting information throughout the analysis of the entire sequence of tokens, but there were two main problems. The first one was the fact that because of their nature, they were sequential processing. So the point was that they were quite hard to be parallelized, so that resulted in inefficiency. And secondly, such models suffered from the problem of vanishing gradient, which seems to be impossible to be overcome. While on the other hand, the transformer takes a one sequence at a time, so it's quite easy to be parallelized. And secondly, it resolves the problem of vanishing gradient by implementing a self-attention mechanism. This is how a transformer is structured. First of all, we have to use an input embedding, which means that, first of all, the sequence of input fed into our model are parsed into tokens by means of a diagram coding tokenizer, which is a form of data compressor. Then each token is converted into a context vector to keep track of the meaning of a token with respect to the others. And lastly, information about position of every token inside the sequence are added to the context vector. Then the transformer has an encoder-decoder architecture. The encoder part has a multi-held attention layer and a multi-layer perceptron layer, while the decoder part has a very similar structure, but here we can observe there's a additional multi-held attention layer. And it was reminding that the multi-high attention layer has the role to quantify the importance of each token with respect to every other token of the sequence we fed into our model. And at the end, we have the classical classification head, because after all, our model aims to classify, so we need a classification head. Here comes the vision transformer, which basically exploits just the architecture part of the classical transformer, but this time is an architecture designed for the analysis of images. So the input should be fed in a different way. So in particular, an image should first of all be subdivided into patches, and then each patch should be linearly embedded and fed into our model. A point that may raise some doubt about the actual effectiveness of a vision transformer is that, as you may have noticed, we didn't use any convolutions, because convolutions are largely used when dealing with images, but this time we are not using any convolutions at all. And this is because we take into account the image-specific data biases by adding this parameter, an extra Lermo classification token, addressing the problem in a very similar way to what happened in the BERT architecture, for instance. So in order to recap, the standard transformer receives as input a 1D sequence of token, while the vision transformer receives a 2D images in the shape of a 3-dimensional data, because we have the dimension of channels. And such images have to be reshaped into a flattened sequence of 2D patches belonging to R n times p squared c, where HW is the resolution of the original image, c is the number of channels, pp is the resolution of each image patch, while n is the number of total patches. Okay, here comes the point when we talk about the reason why we implemented the attention rollout mechanism. So when we use a lot of attention maps, what happens is that information flow gets gradually blended through the analysis inside our deep model. So what happens is that we would like to have a way to quantify the attention flow, and this is what actually attention rollout is. Of course, to use this method, we make an assumption, that is that we suppose that input tokens can be linearly combined through the layers based on the attention weights. This is how we implemented this is how attention rollout should be implemented. So the attention flow is modeled with the DAG, which is a directed acyclic graph, in which nodes are input tokens and hidden bandings. Edges are the attention from the nodes in each layer to those in the previous layer, and the weights of the edge are the attention weights. Also, we have to take into account residual connections. So to do this, we had an identity matrix to the attention matrix, and we renormalized the weight. This results in a new row attention matrix, which is 0.5 the attention weights map, and 0.5 the identity matrix. Okay, to expose what is the model we propose, we shortly describe the dataset. The dataset we had is CT lung, which is made of CT scans of lung cancer patients who have been subjected to biopsy. Each scan is noted about the presence or the absence of a genomic profile. So basically, it is stated whether the AGFR gene is present or not. And in particular, in the subset of the dataset we used, we have that we have 30 times out of 131 the presence of the AGFR. Also, an expert oncologist provided us the segmentation masks. So we exploited them, And it's worth mentioning that, to remind that the aim of the model that we used is to binary classify the scans containing a tumor mass, depending whether there's the EGFR gene. So the model we proposed is made by two parts: a Vit Transformer, which is basically used just to extract attention maps, and a 2D classifier, which takes as input a concatenation between the attention maps generated for a given input and the input itself. This is how we fed the model. So we exploited the 3D shape of tumors by sending as input to our model a single 9 consecutive slices arranged in a 3x3 grid. And internally, the model subdivides each slide into 16 patches, each representing a target for the self-attention algorithm. This is how we pre-processed the input. So first of all, we applied the windowing process using a standard setting. Then we cropped them around the lesion, and we exploited the experimentation masks the oncologist has provided. Also, we resized to 64 by 64 pixel, and we arranged the patches into a 3x3 grid, of course, because they have to be fed into the vision transform. And also that is augmented by applying random horizontal flip and random 90-degree rotation. So during training, our 9 consecutive slices are randomly sampled such that the model is trained to minimize the cross entropy. The optimizer we used is ADAM, and the learning rate 0.001. The loss, of course, had to be weighted about duration between the sample where the IGFRG was present and the sample where it wasn't. And at prediction time, we used the whole sequence, the whole CT scan for prediction, creating a list of patches image, each containing 9 slices using a stride of 2. Each image of the list is independently classified, and the final prediction is declared by judging. And those I'm showing are the results averaged across the 5-fold cross-validation. So in conclusion, our experimental results are coherent with current research. They are quite decent, and they they support the thesis stating that there's a long road ahead of us, and there's a large room for improvements in such field. Lastly, I would like to thank my tutor, Concetto Spampinato, and his PhD student, Federica Salanitri, who really helped me through this process, and who really supported me through facing the great amount of obstacles I encountered. And lastly, I would like to thank the Scuola for its great support, and so also I'm so glad today to be here in presence, because it was not sure I would have been there today. So thank you.